Welcome to SciCon 14, Engineering a Cure. Enjoy.
It's always so fun. It's so exciting to see everyone here. I'm thrilled to have David Baltimore and Feng Zhang. I've been spending a week just cramming information about genetics, and it's been such a pleasure. And I just wanted to start with you, David, and ask you a little bit about your history. You've made these incredibly influential contributions to immunology, virology, genetics, and cancer research. And I'm just wondering how, like, a nice guy from Queens ends up changing the human genome.
We moved to Great Neck. Got out of Queens.
And Feng, no.
I had the notable pleasure of being at the Jackson Laboratory in Bar Harbor, Maine, when I was a high school student. They had a program for high school students there. And I discovered experimental science, working on little kids' experiments. And I never turned back. I said, this is how I want to spend the rest of my life, discovering things, working at the edge of knowledge. And I could have thought, but I didn't, riding the wave of molecular biology that was going to crash in front of me. I have had the great pleasure, though, of running with that wave for some 60 years, and it has been enthralling, every bit of it. I'm lucky enough to have worked in a number of different areas and to have made some discoveries that had broad implications, particularly the discovery of the reverse transcriptase, which was in 1970, which is a long time ago.
So you received the Nobel Prize at the age of 37,
Yeah.
right, which is very young. And
I didn't think so at the time.
you didn't think you were young at the time? Well, you know, now you can cure aging, so we'll get to that. The citation was for understanding the interaction, discoveries related at least to the interaction of tumor viruses and the genetic material in the cell. And this led to unbelievable discoveries, many, but one of which was the understanding that cancer itself was a genetic disease. It's kind of amazing to realize that discovery was made so recently, really. And what is that? I mean, was it a eureka moment? Was it that glorious moment where you just suddenly realized, or was it kind of a slow growing revelation?
I mean, if you look at the discovery of the reverse transcriptase, which is given credit for establishing that cancer was a genetic disease, you had to extrapolate a number of things to get there. And those needed proving, but they were proved very rapidly thereafter, that a virus actually inserted its DNA in the host cell and that then it was transcribed out of the host cell's genome. And once we understood that, we could say that genes cause cancer because we were integrating new genes into the cell. However, we hadn't demonstrated that, and it was really Harold Varmus and Mike Bishop who demonstrated that there was a particular gene that was carried by the virus into another cell, and that in its mutated form, which was carried in the virus, it caused cancer. And that was really the key that broke open the whole field.
It really shook up the field. There was a sort of classic paradigm in genetics. We'll pick it apart a little more, what a lot of these terms mean, but this classic paradigm, and it really overthrew this, creating a lot more momentum suddenly. I want to ask you, Feng, because I know I've been reading about you a lot, and you have many titles, it seems like. Neuroscientist, molecular biologist, synthetic biologist. So you also have a very broad range of topics that you were drawn to, and I'm curious about how you started in science and how you ended up in such a broad range of fields.
Sure. So I was born in China and moved to the U.S. with my parents when I was almost 12, and we landed in Des Moines, Iowa. And
the hub of science and scientific thinking.
And,
Rationalism.
and it was a fantastic place to grow up. It's having moved to a new country, people in Iowa really just welcomed us with open arms, and the teachers were caring and looked out for each of the students and tried to provide them with the right opportunities. I actually didn't like biology when I first started. My parents work with computers, and to me, biology was mostly about memorizing things or dissecting smelly frogs and identifying different anatomical parts. But when I was in middle school, I think it was either 7th or 8th grade, I took a Saturday enrichment class. So these were programs the school held for kids on Saturdays so that they can go, or maybe parents just want to get them out of the house so that they learn something. And one of the classes was on molecular biology. And I had no idea what it was, so I decided to take it. And there, I really had my imagination open. Among many things, they showed us documentaries like Jurassic Park. And, and at the same time, we also did experiments like extracting DNA from strawberries or running a gel or making bacterial cells resistant to drugs. So those are all really interesting. And from that point on, I kind of thought biology is not just memorizing things, it's something you can tinker with and build. And that was something that captivated my attention. And since then, I've just had great teachers and it was a great experience.
Well, you also, at a very young age, if you literally didn't do another thing in biology, have already made so many pivotal contributions. That's not a question, I'm just saying that to be nice. So I did want to kind of talk a little bit about molecular biology. Oh, I did have a DNA slide. I guess it didn't make it in. Pretend we're looking at the double helix. We'll talk about this in a second. When I was studying a lot of the molecular biology and the classic story of DNA as the double helix, I was struck by how inanimate all the participants are in this story. It sounds, they're just molecules, and they're just chemical bonds, and there's just biology. Oh, good, you're going to disagree with me, that's even better. And it just sort of stunned me. How do we get from this description of these organic molecules interacting through electrodynamics and chemistry to life? That just seems like such a tremendous leap. Does it still amaze you how to make that transition?
Yes, it still amazes me. But it's backwards. We first wondered about life, and then wondered about how molecules could create life. And that's actually been my whole existence, is trying to understand how molecules can create aspects of living things. Biology is chemistry, unfortunately for a lot of people who hate taking organic chemistry. And that's what makes us all tick. And the whole story of the DNA double helix is a story about each atom in the DNA molecule, which contributes at one point or another to the coding potential, to the various codes that are found in DNA, various ways that DNA influences the behavior of cells. And so it's the most intimate chemistry in the world when you're talking about biological chemistry.
And yet it doesn't sound alive. Like, here's a picture of a virus. Is that a virus? I hope so.
Well, it's sort of like an idealized virus.
Is a virus alive?
Oh, that's a question that students love. A virus, as you see it here, is doing nothing. It's just sitting there waiting to find a cell that it can bind to, go into the cell, and take over that whole cell. So there's sort of no question that it's alive when it's inside a cell and controlling the cell so it makes more of itself. That's what all living creatures do. But there's a lot of question when it's sitting up there on a slide, whether it's alive or not. And it's all a matter of definition. There's nothing really deep in that question as far as I'm concerned. You can call it alive, you can call it dead. It's matter which has potential, and the potential is to have huge effects on living systems.
Well, isn't it Carl Sagan's quote? I don't know the definition of life, but I know it when I see it. Do you?
That's sort of also the definition of pornography and a few other things.
Yeah, I might have gotten those two things confused. Sorry. What's your feeling about the emergence of life from this sort of what feels like this inanimate process?
Life is complicated. Well, at the same time, life is also amazing. I was just talking with Dr. Baltimore's wife during the break before this.
Who is also a biologist.
Yeah, and talking about grandkids or kids. And if you look at the biological program that allowed them to develop, it's really amazing. There's so many parts. There are at least 20,000 genes, and each one of them can give rise to many more proteins. So the complexity of life is enormous. Yet, for every single one of us, this biological program carries out and gets executed with really high precision. Rarely goes wrong, and that's amazing. That, to me, understanding that program and how it works and being able to fix it to make people live better lives, that's really interesting, and hopefully we'll be able to do.
So viruses loom large in this conversation, and we all know, obviously, the deleterious effects of viral infections, but they've been put to use as well. So when I was listening, David, to some of your descriptions of how a virus invades a cell and takes over the genetic functioning, recodes it, actually recodes the actual DNA of the cell. I was so fascinated by that, and I was wondering, is there some ecological advantage to viruses, or is it just this brutal war between us and them? Is there some sense in which a virus has a positive role on the earth, or is wiping us out like?
I've always wondered that if we got rid of all viruses, would there be an ecological consequence? Well, I'll tell you one place where I wasn't thinking about when I was thinking about that, but where the effect is enormous, and that's out there in the ocean. The ocean is full of bacteria. They're constantly dividing, constantly growing, and constantly being killed off by viruses. And if you take a bucket of seawater and centrifuge down the material in it, and look in an electron microscope, you just see a myriad of viruses. It's amazing. Many of them we know nothing about. They've never been cultured. They've never been, the genes in them have not been identified in terms of what they do. But they maintain the bacteria at a significant but non-lethal level. While at the same time they are killed off constantly by sunlight, by other things. So I think viruses actually serve a very important role in the balance of organisms on Earth, and we're a tightly interwoven set of organisms on this Earth, and if you take away any big part of it, you're going to have a big effect.
So do we know if viruses all have an evolutionary common ancestry, or is nature making them all the time and we're constantly dealing with completely new structures? Feng, do you have a sense of that?
There are different forms of viruses, certainly. And some viruses are based on DNA. Some have RNA as their genetic information.
Can you give us the brief one-line distinction between DNA and RNA?
Sure. So we are encoded by genetic information, and that genetic information is called DNA. But in order for that genetic information to get turned into something useful for ourselves, it has to be made into protein. And the way that the information goes from DNA to protein is through an intermediate messenger, and that's called RNA. So it's the cell converts DNA into RNA and it uses the RNA to make protein. And so viruses can carry either form of genetic information, either in the form of DNA or in the form of RNA. And these can have different things, different ways that they're organized. But just going back a little bit, we, over the course of evolution, have been exposed to many, many viruses. And there's one interesting study that came out maybe a couple months ago, where we and viruses have a symbiotic relationship. And so sometimes our body, our cells, have harnessed or taken advantage of some of the viral components and made them useful. So for example, there's this protein that they reported, it's called ARC. It's a protein that's made in our brain cells. And this ARC protein is actually derived from one of these retrovirus proteins called GAG. And so, who knows where along the path of evolution, how many billion years ago or hundreds of millions of years ago when this adaptation happened. But animals have converted this virus protein into something that's potentially useful and intrinsic to the biology of an animal. So this kind of borrowing happens back and forth. Viruses borrow things from cells that they infect, and the cells they infect borrow things from viruses. And that web of activity is life.
Mm-hmm. David, you mentioned the discovery of reverse transcriptase, I hope I'm saying that right, which is an enzyme that transcribes DNA from RNA, and it was long thought that it could only go in the reverse direction. And along with that discovery, there were many, obviously, significant consequences and implications, but also you identified an entire class of new viruses, which were the retroviruses, of which HIV is a very fierce example. And I know you've done a lot of work on HIV. And I just wanted to pause for a second to think about those statistics. So I watched a talk, I don't know if you have the latest numbers on you, but I watched a talk in 2005 where the numbers were already staggering. Do you know what the numbers are now, the statistics of contraction and...
I know some of the numbers, but I can't actually remember them terribly well because they're all basically the same as they've been for
So in 2005, I was quite stunned. It was 45 million infections with HIV, 25 million dead. There were 5 million cases a year acquiring the disease and 3 million dying a year. I mean, that's really staggering. So what is it about HIV that is so difficult to control as an infection or as a virus?
Back to sex. We transmit HIV, and the reason people get it is that it's been transmitted from another person who has it. That's the nature of viruses, to be transmitted from one organism to another. If we found a way to interrupt that with a vaccine, for instance, then we could really protect people against getting HIV. And I think we could put the genie back in the box. We could get rid of the virus. I don't see any other way of doing it. Many people have spent long hours trying to make a vaccine against HIV, and no one has succeeded. I'm just hopeful that one or another of the ideas that are being tried now will lead to a vaccine. Because without that, I think those numbers will just keep growing. In the United States, we have 50,000 new cases of HIV infection a year. That hasn't changed in the last couple of decades by now. It's down a little bit now because the drugs are actually having an effect on the transmission. But it came out of monkeys, through chimpanzees, to humans, and we haven't been able to get rid of it.
So do a lot of the viruses that cross the species barrier, are they particularly virulent?
Yeah, in general, they're viruses that haven't evolved with that species because they just jumped into them. So they're either very virulent or they're not virulent at all. So Ebola is a virus which is very virulent for somebody who gets it, but it transmits very poorly. So mostly it burns itself out. The last epidemic that we had might have actually exploded, but was finally controlled. But HIV has found fertile soil in humans, and it only took a few mutations of the monkey virus to turn it into a human virus. It did take a few mutations. But then we have flu, and flu comes into the human population, and because of the nature of the structure of the virus, it keeps infecting us year by year with new strains. So there's a story behind each virus, and each story is different, and each story is fascinating to people like me. And each virus is its own biology.
Now, the fact that a virus can penetrate, let's say, a human cell for the sake of argument, and alter the DNA makes you wonder if it could be put to use as well as a vehicle for gene therapies. And I understand that you've both thought about this and worked on this. That's kind of a fascinating prospect, that you use the virus to cure the disease from the virus. And with HIV, do you think that there's still that kind of viral gene therapy is still viable?
Oh, yes. I mean, we're using a stripped-down HIV to move genes around in experimental organisms and even now in humans. Once you strip it down, it's not going to cause any disease of any kind. We can be sure of that. But we can put in genes. Now, putting in new genes into cells, we can't be sure what's going to happen, but that's another story.
And then I think this, if I understand correctly, that's still a very difficult engineering problem. And
no, it's a very simple engineering problem now. Anybody at the stand over there could carry out the recombination necessary to put a gene into a virus and to get the virus into cells.
Well, that brings us to CRISPR, doesn't it?
Right.
So maybe we should talk about CRISPR. And I actually borrowed this movie from Feng. This is one of your animations illustrating this process. Can you talk us through this? Here
Sure.
we have.
So here we'll have a blue glob, and that's called that's a protein called Cas9, and the red strand is a strand of RNA, and the RNA and this blue glob work together to be able to recognize the DNA. DNA is this double-stranded helix. When the DNA is opened up, then the RNA can pair with the DNA, and when the RNA and DNA forms a perfect pair, then the DNA gets cleaved. So it gets severed into two pieces. And so this severed DNA, this DNA break, is what triggers or allows us to be able to edit the DNA. And so what just happened over here is that the two ends got stitched back together, but a small error has been introduced, and that error is something that's useful for inactivating a bad mutation. Alternatively, when you have these two broken ends, you can give it another piece of DNA that should be floating around. Somehow the
I
movie
couldn't
doesn't
get the
pause.
rest of it. Yes.
Okay. So then when you give it a new piece of DNA, a synthetic piece of DNA, with a new DNA sequence, then during this repair process, that new sequence can get very precisely incorporated into where the DNA break has happened. And so those are the two ways that we use these CRISPR systems to be able to edit DNA in cells.
So now it's become very cheap and easy to edit cells, but it's actually a naturally occurring phenomena. CRISPR, I mean, it originated, bacteria made it up before we figured it out.
CRISPR is a bacterial immune system, very much like how we have an immune system. Bacterial cells use CRISPR to fight off viruses. And it uses this RNA mechanism to remember viruses that the cell has seen before. It will actually take a little piece of that virus and keep it with the cell, with the bacterial cell, so that next time when the virus comes, that piece that was obtained from previous virus gets converted into a new RNA, and that guides the enzyme to recognize the virus and destroy it. And you can take these components, engineer, optimize them so that they can work in human cells. You can program it with a new piece of RNA, now not to recognize viruses, but to recognize a faulty gene in the human genome. And then the RNA can take the enzyme to that faulty gene, cut it, and then allow you to repair it.
So this naturally occurring mechanism keeps a vaccination card, is how I understand it, in the actual DNA of the bacteria. And so given bacterium that's been exposed to a certain virus can pass it on to the next generation. And it would be good if we had something like that. Why don't human beings have that kind of...
Well, so this ability to remember the infection that the cell has seen before is called adaptive immunity. And we have adaptive immune systems as well. It's part of our immune system that have antibodies, and these antibodies can recognize viruses or bacteria that invaded our own body, and then be able to keep that memory so that the memory passes on. The only thing is that it doesn't transmit to the next generation. So our children don't necessarily have it. But there is some that gets passed on.
So it was known for a long time that this system existed in bacteria. And then, no, were you saying no, David? Was it not known?
Not a very long time.
Like 2 decades? No?
Well, more like a decade, I think, before you got to work on it.
Well, my understanding was that it was known to exist naturally, but the idea of being able to adapt it into the human genome was the big advance, being able to discover that you could synthesize guide RNA to take things like the Cas9 enzyme, find a particular spot in the human genome and edit it. And the work that you've, I understand, done a lot of has to do with making that viable in human cells. And so what was the big obstacle that you overcame to make that possible, to begin to edit, actually get it into the cell and make it actually function?
So this CRISPR system typically it exists in a bacterial cell. And bacterial cells and human cells are pretty different. Bacteria have their genome is organized in a simpler way relative to the way that a human genome is organized. And also the way that all of the chemicals and ions and temperature in human cells are also different from a bacterial cell. And so in order to get a bacterial component, take it out of bacteria, put it into a human cell, is like taking something from planet Earth and putting it onto planet Mars and hope that in that new environment, low atmospheric pressure, much more UV radiation, gamma ray, can still survive and work. So you have to engineer the enzymes so that they can get produced in human cells, they can fold properly so that they can form the right shape so that they can go into the DNA of a human cell and start to find the faulty gene and recognize it, cleave it, and all those different components you have to one by one work through. And that's kind of the process to develop these bacterial systems as a technology for treating human disease, for example.
So human disease is the big prize in a lot of these studies and a lot of these ambitions. And what are the actual functioning trials that are being done now to therapeutically, they're actually being in practice on human beings?
There are two kinds of use of CRISPR. One is to modify the behavior of errant cells of some kind in an organism. And that wouldn't be passed on to their children, but it can help people in a whole variety of ways. The other is the actual alteration of the human genome so that you get rid of a faulty gene. Or some people think you can put in genes which have attractive properties. But let's just worry about faulty genes for a moment. The only place you can get access to those genes is in an embryo that has a very small number of cells, or maybe only a single cell. And then you can alter the genes in those in a single cell, ideally. and it can give rise to a whole organism, and that will not have the defective gene anymore.
Has that ever been done completely
Well, it's
to
been
adults?
done in animals extensively, but no, it hasn't been done in humans, I think. Who knows what's going on? But it will happen in humans because people want to have children that don't have passed on to them deleterious genes. And there are about, 3,000 of those genes known now in
And the
humans.
point being that they're single genes that need to be replaced, making
Right.
it more manageable than a whole complex.
That's right. And that, we're still not doing that because we're not sure of the safety of the CRISPR-Cas9 complex, and because we're not sure that we can modify only a single cell without there being mosaics, embryos that have some cells modified and some cells not modified. And when you have that, you can't be sure what's going to get passed on to the next generation. And so you haven't really done what you set out to do.
So I believe it was a Chinese group that actually did the first CRISPR-Cas9 edits on embryos that were surplus embryos for fertilization clinics and were considered non-viable was one of the criteria they used for the embryos, meaning that they could not be brought to fruition. And they saw a lot of other cuts and edits that were unintended. Do you think that's because of the particular embryos they were working with were already sort of genetically complicated, or do you think that that's just a problem with this particular bacterial mechanism that it was cutting kind of freely and maybe not so precise in general?
Yeah, that's definitely one way to put it. I don't think anybody expected that experiment to give a nice, clean answer. It was still, we hadn't perfected a lot of the aspects of the system, and so it was premature. But it was really, to my mind, important because it showed us that there was a reality to the worries that you might have. And I think that has gotten the field a little soberer, and people are now working on the details to try to make them as perfect as they can be so that we don't have, quote, off-target effects. And I think we are getting there, although actually you have to prove that for each individual guide RNA that you use, so each individual disease. But if you can get one that you've tested out that doesn't have a lot of errant activity, then you could get a perfection of modification that we need to use it in humans.
Feng, do you feel confident you'll be
Feng
able
knows
to
more
use
about
it
this than I
very?
do.
Are you confident it will be a very precise system and that you won't leave a patient walking out the door worrying that they're going to come back with unexpected results like horns or something? Like, how do you know if you're unsure what all the edits are going to be? How well do we really understand these things?
This is one area that's undergoing really rapid development. This specific effect is about the specificity or the precision of the edit. The human genome is vast. It's 3 billion letters long. And so if you are programming this little machine to hone into a specific spot, there are 3 billion other places where this thing can make a mistake. And so how precise can we program this to recognize only the gene we want to change and nothing else is the question that we're talking about. So we know the very first generation of the technology can make unwanted changes. Sometimes it will make a mistake, and we want to avoid that. So a lot of work has gone underway. Some of the latest generation of the technology has been engineered, taking this protein, altering it so that it only recognizes what we wanted to recognize and not have enough stickiness for the things that we don't want to recognize. And those systems are now showing to be quite specific.
And you've discovered other proteins as well, right, in bacterial systems that might have advantages over the Cas9 enzyme. Have those been implemented yet?
Yeah, and those are being tested as well for in preclinical models to evaluate what's the most precise but also most efficient system that we can use. CRISPR is a system that's pervasive in bacteria. Over 60% of bacterial cells and over 90% of a more ancient type of cell called archaea, they all carry CRISPR systems. And so in each one of these systems, there's a different type of CRISPR. And so because of that, there are thousands of different Cas9s. And these different ones have subtly different properties. Some are a little more specific, some are a little less specific. And so one way to improve these systems is to analyze the diversity of these proteins out there from different bacterial species. And some are turning out to be quite specific.
So I wanted to actually ask, in connection, these are the SciCon patches, by the way, due to Andrea Lauer. Aren't they awesome? They're very clever. It's a medical pill with DNA as the ingredient. So I'm wearing mine. So with this promise of, what was it? Would you like one? I'll get you one later. With this promise of gene therapy medical benefits comes, pharmaceutical companies, comes the attempt to actually make this something that can be distributed. And that comes out of academia now. Now we're leaving this, dreamy land where we just investigate in the laboratory and we just pursue our blue skies ideas. And all of these biotechnology companies have cropped up around these, the promise of the gene therapies. And I believe you both have biotech companies that you're affiliated with. And how do you feel about that transition from the openness of academia, where the funding is publicly funded and where it's supposed to be for the public good, to something that becomes more monetarily driven, maybe, and where patents have become a big issue? How does that, is that a crisis at all for you, or is that just the natural extension of what has to happen next?
Well, if you go back to the very first biotech companies, Genentech and Biogen and a few others, as they developed, I asked myself that question. And it seemed to me that if the government stepped in and made sure that the people weren't making ridiculous profits on this, and that they were working for the public good, using the new biology techniques, that would be fabulous. And I talked to a number of people about that, people who've been in the corporate world, which I had not. And their answer was uniformly, you can't ask the government to do that. In fact, there's nobody you can ask to do that. You have to let the play of the market work in order to get these advances that are coming out of universities to the patients. And I've come around to believing that. But it means you inherit all the bad things of corporations along with the good things. And so you have to work to try to minimize the bad things. And I've had the opportunity to work inside a number of corporations. But you also have to be realistic and understand that there's a whole system of funding of corporations which you have to work with if you're going to actually build a pill that makes a difference in people's lives.
So I've been pretty fortunate to be involved in the development of CRISPR technology. And CRISPR
and you've secured some of the very important patents, which is exciting, but must feel complicated. I didn't mean to cut you off. I'm sorry.
And CRISPR is a very foundational technology. It's useful in research, in drug development, in developing gene therapy, and also in agriculture, many other applications. And so for a fundamental technology like this, what I think, and also what my colleagues agree with me, is that it should be kept open. It should be made accessible to as many people as possible around the world, so that we can really accelerate science and research across the board. And so one of the things that we received was patent rights for CRISPR technology for genome editing in human animal cells. And having that patent made it possible for us to try to make this publicly available by ensuring that this is non-exclusively provided to as many parties as possible so that all of them can have the rights to use it. If we didn't have the patent rights, then someone else may have the patent right. And then we don't really know what their philosophy will be for a technology like this. Maybe they will want to keep it closed. In the genome editing field, there are earlier technologies like zinc finger nucleases, which were where the rights and the know-how were proprietary. And that wasn't so accessible, and it was expensive for researchers to get access to. But with CRISPR, I think it's really great that we were able to make the technology open and accessible so that people around the world are now using it.
To academic scientists in the university context.
And also commercial developers. Commercial companies can come and get a non-exclusive license to the CRISPR technology, whether it's in the agricultural industry or in pharmaceutical development. And all of these opportunities, I think, is enabling researchers to do good things.
Now, some of the things that people want to do are, some of them are kind of whimsical and fanciful, and others are more serious. So gene therapy to improve the human condition. I mean, there's also people making pets, very tiny pigs, dogs with special coat patterns. And this is being done. And I think, David, you sort of laughed, well, who knows what people are really doing? But if it's that cheap and easy, you really don't have a handle on what people are doing. Yes, it would be great to cure cancer, prevent HIV, cure leukemia, have biofuels, spectacular things outside of the medical realm, have wheat that survives droughts and winters, and really make a positive and benevolent impact on the world. But then there are these other possibilities that seem a little bit too easy to do, too difficult to regulate. Do you have concerns that anybody can do anything?
I would if that were true. But if you're going to try to change a gene in a human, that's not simple. And in particular, if you're going to work with human embryos, it requires a whole technology of in vitro fertilization and then analysis of embryos. Those things aren't going to happen in people's garages. If you want to modify a gene in a yeast or a bacterium, yes, you can do that in your backyard today or in your garage. California, it doesn't rain, so we don't even bother with the garage. But those are not dangerous things, to my mind. The things that happen in agriculture are a little more complicated because there's been such a negative view of GMOs, of genetically modified organisms, in the food chain, whether it be animals or plants. And in the United States, we've been a little more accepting than people in Europe, but there's a big negative force in the United States, too, against GMOs. I think that's put a damper on the development of the use of genetic modification in plants in a very unfortunate way, because our world needs better plants as foods. We have more and more people on this planet, and that's not going to change, even if you might want it to change. And they have to be fed. And so we have to find ways of using this technology for the modification of plant crops and animal husbandry, animals that are husbanded, whatever the word is. And then we can see the full power that this technology has on the lives of the world.
Now, let's say it must be relatively simple, maybe it's not, but to modify the genome of mosquitoes, for instance. There's, not that we know how to do this exactly yet, but the idea being you could prevent mosquitoes from transmitting Zika or malaria or Lyme's disease. And yet there's also a concern that once you release that, you don't really understand the impact ecologically, and that it's not something that can be reversed easily. But Feng, do you share that concern?
So what you're talking about is something called gene drive. So you can engineer a mosquito so that when it breeds with other mosquitoes, it can transfer that genetic instruction. If that genetic instruction is you can't breed anymore, then that species after a few generations can go extinct. So that particular technology, gene drive, I think is something that we should be thoughtful when we're developing, because if we're talking about changing large populations of a given species, it's hard to predict and hard to imagine what the ecological impact of that large species modification will be, and so I think that particular application is something that we should definitely be careful about.
Wasn't there a call to make sure that it could be reversed before a gene drive was ever applied? But there's no actual regulations, are there? Or are there just agreements among scientists to use ethical thinking, and can we trust the entire world to adhere to these principles?
So people are working, so this is an area that there's quite rapid development as well. Researchers are working on ways to be able to regulate it, to be able to switch it off, to be able to reverse it. And those are all things that are underway. And there are also a lot of discussions and working groups that are very much like with gene editing that have convened together to first establish a common understanding, and then from there try to think about ways to be able to guide the development of this technology.
Isn't that how we feel, though, also about things like nuclear energy? And some of our neighbors don't adhere to the same principles that we'd like them to with dangerous technologies. What makes us feel confident that other countries which have different principles or are driven by different ideals won't make kind of chemical weapons or biological warfare out of simple things like CRISPR edits?
I think if you think about gene editing and so forth, first and foremost, on the scale of things, what's easy to do might be to treat a human genetic disease. And even for those very easy things to do, we can treat sickle cell disease today.
We cannot?
We cannot. We don't have a good way to treat cystic fibrosis. We don't have a good way to treat hemophilia. And even for those very easy things, we can't even get there. What I'm really worried about is how fast can we get to treating these diseases, to get these things that can have a very tangible positive impact on patients' lives done. And we can't even get there yet. So that's what I think we need to focus on.
But that's you. You're a nice guy.
Yeah. So let me talk about that for a minute, because I've organized one, and now there will be two international conferences on gene editing. That in gene editing in humans is the major focus of it. And the reason that we've organized these meetings, and the reason that they are as international as we can make them, the next one will be in Hong Kong, is just the reason you ask. That we need a way of coming to an international agreement, and it's got to be done driven at least by the scientists, because we're the ones who really know what it can do and how it works. And so we have a large number of scientists, but we also have ethicists and newspaper people and lots of others, because we want to share our thinking with a wider public as much as we can. So the goal of these meetings is to come to an international understanding with such force that countries that might think about violating international norms would think twice. We can't be sure. You can build a scenario that that things were happening behind some iron curtain, and we can't know about it in the general public, and that is certainly possible. And I don't think there's any way of guaranteeing progress in science without having to think about these very difficult international issues. But we're working on that.
How long away do you think we are from something like an international agreement?
Well, we're closer than you might think, because I found in the first meeting, which was in December of 2015, that there wasn't wide disagreement, and that if in any of the countries, that were represented at that meeting, anybody did something errant, and I won't even try to define it, I think there'd be huge pressure within that country from governments, from journalists, from others, to stop that. And that's what we've got to build up. We've got to build up this kind of international pressure so that if somebody tries to do something, they're cut off.
I promised people that we would talk about death. So I have this, there's applause for death? Okay. So I have little...
That should read autonomous there.
Yeah, so this is part of the animation, David, where you're narrating. This is brought to us by the group Massive, and it's a beautiful little animation. We're not going to hear the sound, but we're just going to show it while we talk about the idea that I know George Church has discussed, very ambitiously trying to edit the DNA to either end or reverse aging. Is this something that you think is conceivable, or is this just a dream? I guess anything's conceivable at this stage.
Well,
I mean, are we literally talking about immortality as a realistic end point of the CRISPR ambition?
It's very hard to use CRISPR on anything somatic, anything in our bodies, because you've got to transform so many cells. It means you've got to have access to all of those cells. They've got to stay there and in their modified form. So this kind of thing in that picture of taking the DNA out of somebody, modifying it and putting it back, you can do in one cell, you can do in 10,000 cells. Whether you can do it well enough to actually turn around a disease or a bad gene I wait to see.
What do you think, Feng? You're young. What do you care?
Well, I think in terms of aging, there are many biological questions, and the mechanism how aging works, we have no idea about, and it will take a long time to research.
We once had no idea about CRISPR either. So it's just a matter of time, in other words, or is it not? Yeah. Do you think that there's some fundamental obstacle, or isn't just a matter of time? Someone ambitious enough, interested enough, deep diving in that particular topic will make progress.
It will take a long time.
I thought we've seen reversal. It would take a long time. Forever is a very long time, especially the bit towards the end. Yeah, that's a joke. It's not my joke. So I understood that they took aged cells from mice and could actually revert them to stem cells, and then revert them back, let's say, to skin cells, which was what they were originally, and they're younger. They've erased the aging process. Hasn't that happened in the lab?
Not exactly sure which study you're talking about. But
I might have made it up. George Church told me about it. He's your colleague.
Yeah,
go back to the Broad Institute.
Let's imagine that it did happen.
Yeah. So, do you think it is just a matter of time, David?
I think, yes. I think it's just a matter of time. I think we have now an army of very skilled people trying to understand all of the effects of all the genes in the body. Aging is controlled by genes in ways that we don't understand, but it's too reproducible for it to be a consequence of the environment, which is what a lot of people like to believe. Radiation or something you eat or whatever. It's none of those things. But it's very hard to understand aging. For one thing, there's no experimental organism that ages the way we age. We age now over 100 years, roughly. The organisms we work with in the laboratory live 2 years, mice. They age over those 2 years. They get gray just the way we get gray, so there's clearly a homology there. But I'm sure that our aging process is much more complicated than a mouse's aging process. And we still don't understand it in a mouse at all. So it's a long time before we understand it. But I do believe we have the tools today to understand anything in the body. And that includes the brain, which is the hardest thing to understand.
Mm-hmm. So we discuss all of these benefits to humanity, gene therapies, medical therapies, things like biofuels, helping bees maintain their hives maybe through genetic editing. But is immortality and not aging one of those good things? I mean, aren't there some kind of really obviously negative consequences to nobody dying?
Yeah, the first thing to say is aging is not a disease. It's a natural process. And so if we want to muck with it, we could decide to muck with it, but it's not going to be like getting a universal flu vaccine, something I think we desperately need.
Mm-hmm. We really should open for questions pretty soon, but I was going to show another clip from the animation, which you can find on our website if you want to see the whole thing with the narration. And this sort of refers to the idea that we'll try to do something through gene editing to make somebody stronger or healthier, but we won't always understand the implications. And David, this was coming from your conversation where you were saying we can't always foresee what's going to happen. We might actually radically alter the human genome in a way that we can't predict. And don't be too distracted by the animation. And I wonder if, when people talk about things like AI, there's all these scares, there's this kind of fear-mongering over AI. We've had conversations here with Jan LeCun, the computer scientist, about that, and that AI will get to a point where they take over humanity and become our successors, and that will be our only role in evolution that's of any significance. I wonder if this isn't a faster, more powerful way to invent our successors in some way. There are clearly discussions about designing people for certain tasks, making astronauts with lighter bones or more resistant to radiation or needing to eat less so that they can be on Mars for 2 years without supplies. Are you concerned about the fact that we're basically interrupting evolution to the extent that this is a totally new chapter? No longer natural selection, but human selection?
Yeah, I don't have to worry about that. That's somewhere out in another century, perhaps, or certainly in many decades from now. But do I doubt that it will happen? No, I don't.
You don't doubt that we will invent our successors?
I mean, our curiosity as scientists is going to lead us to understand all of these complicated things and to make the tools to allow their modification. I'm very sure of that. And we're going to have to decide what we want and what we don't want. Now,
people don't always make good decisions.
Well, they sure don't. Right now, we make babies by a genetic lottery. It's who you happen to marry or screw around with. And it's it's a complete lottery. And you can come out with a wonderful result, you can come out with an unfortunate result. And we're not going to be able to control that until we get, and this was Aldous Huxley's perception years ago, until we get a country, until we are a country, where there isn't freedom of reproduction, where there's such government control that the government decides who you mate and what kind of genes are put in. I don't want to live in that world. I don't even want to see that world happen. So I think the maintenance of democracy, something under real attack today, is the most important thing we have to prevent those kinds of terrible thoughts from becoming realities.
Feng, I'm nervous of your optimism. I'm nervous you're going to be overly optimistic.
I think we need to be humble about life and about nature. So we were talking about HIV earlier, and I'll just give you one example where we need to be aware of the complicated interactions that can happen in cells. So there's a small fraction, maybe single-digit fraction of individuals who are naturally immune to HIV infection. They don't have a gene called CCR5. And so, putting on the designer baby or the baby designer hat, you might think that let's just get rid of this gene.
Yeah, just cut it out.
Yeah, we'll be able to vaccinate the population against HIV. It turns out that when a person doesn't have CCR5, the risk for West Nile virus increases significantly. And so we don't have a West Nile virus epidemic here today, but it's hard to predict what will happen in the future. What new viruses may come up where engineering this genetic mutation would make us much more susceptible to. And so I think it's important to be humble, and it's important to recognize that we don't know what, by perturbing one thing, we might cascade into the entire biological system. So making those changes is something that we should be very careful of and not take very lightly. As a society, that's also something that we'll have to be very mindful for.
Well, I think that's a beautiful note to end the conversation and open it up to Q&A, the idea that we have to be humble in the face of nature, despite the fact that we're rewriting Darwinian evolution with genetic editing. Let's thank our speakers again before we open it up to questions. We have some mic runners. Can we get those mic runners and constantly running them around? Do we have mic runners? Yes? Whoever has a mic. I can't see you very well. Yes, please. Yeah. You're handing them around? Oh, hand them around to anyone who has their hands up. Yeah, here you go.
Thank you. Thank you so much for inspiring lectures on the future application of CRISPR-Cas9. I'm a student from Columbia University. My question is addressed to Dr. Zhang. Is there an opportunity, or can you foresee in the future that scientists or people can use CRISPR-Cas9 or other gene editing tools to edit a human embryo and make every newly born baby to become a perfect genius and intelligence born in the society?
Interesting question.
You were edited, weren't you, Feng?
I got lucky in the genetic lottery.
Yeah, you got lucky.
I think it's going to be hard. Biology is complicated. I don't think you can even define what you want to do, much less know what biological changes to make to effect what you want to have as an outcome. I think that's going to be pretty hard.
Perfect is boring, right? Perfect is boring. What do you think, David?
I think it reflects the fact that you're a Columbia University student. That you think that geniuses should run the world. It's the diversity of human life that allows societies to be built, that allows economic systems to function. If we were all Columbia University students, I don't know what kind of world we'd have.
I do. I teach there.
Yeah, all right.
Who has a mic? We have a mic over here. Yes, please.
Make sure it's on. Oh, is it? Okay, got it. So I had a question either of you can answer. I was really interested in the prospect of the aging process. And as far as I know, it has to do genetically with the shortening of telomeres. How would CRISPR be able to combat that?
Could CRISPR lengthen the telomeres in the cells?
I don't know. Maybe you could figure out a way.
I don't
Now you've got them started. By tomorrow.
By tomorrow. But let's go back to the premise of the question, which is that aging is due to a shortening of telomeres. Aging is due to a lot of things. Shortening of telomeres is one effect of aging, as far as I know. People tried to show that it caused aging, and it's very difficult to show that, and it really depends on the organism you choose. Because the first place you go is to mice, and you shorten down the telomeres, and they have very, very long telomeres, and so it doesn't matter for a couple of generations. Aging is too complicated to say one thing is causing it, as far as I'm concerned.
Yeah, CRISPR is just a tool. It's first and foremost a research tool. It's useful for doing experiments and accelerate our progress to understand biology. Certainly not going to be able to be used for everything under the sky. I think extending telomeres and asking questions like, can we use CRISPR to solve problem A or use CRISPR to solve problem B? That's probably not a lot of them are probably not so relevant.
Who else has a mic now? Wherever that mic went? Somebody has it? Go ahead.
Hi. Hello? Number 2?
Yes, I think. Yes, please.
Hi. So obviously genetic engineering is an incredibly powerful technology with consequences that span potentially tens of millions of years even in the environment. My question is related to potential non-consensual genetic engineering, particularly of humans, whereas there are these enormous incentives with genetic engineering for instance, a for-profit corporation where you might engineer your own consumers, where you might create a genetic affinity for a certain style of logo or a certain genetic dependence on a particular class of product. So what potential defense mechanisms for such non-consensual, perhaps large-scale engineering of people surreptitiously could potentially be developed to protect ourselves from those kinds of attacks?
I don't think there's any way you can do this surreptitiously. The premise is very hard to imagine, because what's required to use CRISPR on the human genome, so it's in an inheritable way, is all of these things I was talking about: in vitro fertilization, the ability to analyze embryos, all sorts of things that go into it. That's not something that will happen non-consensually. Sex happens non-consensually, and that's something we have to control. But I don't see us having such control. And again, it is a matter of democracy, but I don't see us having the kind of control that would be necessary to actually force women to have children that were altered in a particular way.
You agree?
I absolutely agree. I mean, just for the sake of discussion.
I mean, there are dystopian societies where other bad things happen as well. I mean,
Yeah, what if artificial wombs were developed?
Artificial wombs.
Mm-hmm.
Yeah. That's an interesting question. I mean, we don't really understand why the embryo needs to be implanted in the uterus, but we're getting there, and we will understand that. Then can we make an artificial uterus? I don't know the answer to that, but it's interesting to think about, because you're right. That would be an enabling technology for the kind of problem that this gentleman was raising.
They were proposing an artificial uterus, I thought, for bringing back the woolly mammoth also through these techniques, reviving the genes and implanting them possibly in certain African elephants, but not endangering the elephants and using artificial wombs. But so far, I don't think it's been very successful. They're big animals.
We'll hear about
I don't think there are any woolly mammoths out there. I think we might spot those.
We'll hear about them. Right.
Yeah, we'll hear about it. Who else has a mic? Yes, please.
Hi. I had a question about the relationship of like rare diseases to, specifically I'm thinking about progeria. We have all this work talking so much about aging. Progeria, loosely, from what I understand, is this idea of like advanced aging, where someone ages or usually dies by the age of like 16 or 18, and like, but the mind is developing in real time as like a teenager, so forth. So this idea that with the advance of medical innovation and R&D, they're saying that they're looking at progeria to look at aging, let's say. Perhaps there's sort of like a switch in progeria as a rare disease that has implications for everyone else. So I was wondering if there's a correlation between rare diseases and something that is more common, and whether or not this idea of progeria and aging is true or not.
As far as I know, it's true that progeria mimics a much more rapid aging, causes a much more rapid aging process, and people age through many of the stages that are normal human aging, but much faster. So by 20, they're older people. But I don't think that has told us what we wanted to learn. Because it turns out that the genes involved in progeria have to do with DNA repair. They don't, it's not an aging switch. What it is an accumulation of problems that mimic the aging process and do it much faster. But I don't think it's real aging.
Somebody, you can put your hands up when the mic runners are running around. Who else has it? You can just shout out. Yes, please.
If you think of the genomes in aggregate as information, or as we call it in physics, entropy, the whole process of mutations and evolution that's carried out through random mutations, does it increase the amount of entropy in aggregate, as the second law of thermodynamics would suggest, or does it decrease it? Do we go to an increased state of order or an increased state of disorder?
You mean in the gene editing process? Is that what you mean?
Well, the follow-up question would be, how does gene editing impact that? Absent CRISPR and absent any intervention, the process of mutations and evolution, does it increase entropy in aggregate? And then what effect can CRISPR have on that evolution of entropy?
Entropy is the concept that caused all of us who are not in physics to run away from physics. And so trying to use that concept in biology, I'm not sure is perfectly helpful. But you could argue that we, human beings and the organisms on Earth, use energy to counter entropy. And we generate enough energy that we reverse the tendency toward chaos, which, no question, mutation would be. But luckily, at each generation, we go back and start over again. And so all of the entropic, I can't even use the right words, the entropic forces get lost because you die. So in that sense, death is a natural and enriching process for the species. There is a certain leftover mutation rate in the germ cells. There's no question about that. And those things have to be countered, and the thing that counters them is the fact that they give deleterious effects on the organism, and so the organism doesn't live well to breed or doesn't have as many children or whatever. So we purify at every generation our genome, and we need to do that. And if you actually look at the genomes of any two people in here, they're enormously different because each of us has gone through a different lineage of development as an organism and with a different purification.
Can I just throw in there that, the fact that, let's say you have a mutation in an egg which leads to a bird with a particularly ordered set of feathers, I think there's no question that the entropy of the entire system must increase because what goes in metabolically to making an egg and producing it and the energy of the animal that lays the egg and the food that they eat and the sweat and that's usually the situation here. I can't imagine it to be much different than the argument that arranging a stack of cards globally increases the entropy of the universe because of the amount of effort that went into ordering the cards. I don't know, but you guys are the biologists.
I run away from entropy, so I'm certainly no expert. But you should talk to my friend who is a professor at MIT also, Jeremy England. He's been studying how life comes as a result of thermodynamics, how sort of going toward having more entropy actually is the process that gave rise to highly ordered systems and life. I'm no expert on that subject, but I think since you're interested, you should really look him up. Yeah.
Do we have one? I think we only have time for one more question.
I have one.
Yes, please. Excellent.
I know that CRISPR is based on the Cas9 protein, and I know that people are discovering other Cases out there. So I was wondering if you could discuss what are the different kinds of editing possibilities with different Cases that exist.
Yeah, great question. So CRISPR is a whole category of bacterial defense systems. It's like fruit. And then there are apples, bananas, oranges, and so forth. And Cas9 is like an apple. Cas12 is like an orange. Cas13 is like a banana. And so they have different flavors. Cas9 can edit DNA. Cas12 can also edit DNA, but in slightly different ways. And then Cas13 can edit RNA. You can modify RNA. And Cas13 can also be used for detection of DNA or RNA molecules. So you can use it to diagnose Ebola infection or Zika infection. And so that just underscores how diverse CRISPR systems are. And so by fishing out all these different things, we'll be able to probably develop many more capabilities.
I'm actually going to take the last question for myself. I want to ask you, Feng, and then you, David, what you're most excited about on the horizon of this subject.
I'm excited about a whole new wave of therapeutics. If you think about treating diseases, we start, a lot of drugs have been built out of small molecules, chemicals, and then the next wave came protein therapeutics. And now we're really entering into a new wave of more designed therapeutics, gene therapy and cell therapy. And I think in the coming years, decade, we're going to see many more new drugs developed based on our understanding of the biology and the disease process. And those will work more potently and also much more safely. So I'm really excited about that.
And you, David?
Well, there are a lot of things that excite me about what's going on in contemporary biology. And I myself am probably not going to contribute to any of those. But what I'm very glad to see is a maturation of the use of gene therapy, where viruses are harnessed to bring genes into cells and to allow those cells to do things they couldn't do before. That has been a dream for 50 years. Really, ever since we understood retroviruses, we've been thinking about how do you use a retrovirus to bring a gene into a cell. But only recently has it really been done. But I think we're going to see more and more uses of it. It's not easy, and the FDA is very careful about it, so you have to do things in the right way. Really, only a company can develop gene therapy methods because universities just don't have the stick-to-itiveness to do that. But that is very exciting to me because it's something that I dreamt about 50 years ago and have tried to help move along, and now we're really seeing it happen.
Well, I think it's exciting to hear that optimism, given how much you initiated a lot of that entire subject. But I think you're both incredibly naive, frankly. No, I'm just kidding. I think you're both extremely optimistic. I have a little bit more of a vulnerability towards dystopias, but I love the idea of ending on a utopian vision which somebody's snickering, where we really do have this sort of ability to come together and make something positive happen. It's been such an honor to have you, Feng, and David, thank you so much for being our guests.
Thank you.